In 2007, a fisherman off the coast of Grand Cayman spotted something strange floating at the surface. It wasn't caught, it wasn't struggling, it was already dead. He pulled it out of the water, small fish, about the length of his hand, and then he noticed the stomach. It was stretched out beneath the body like a balloon, thin, swollen, almost see-through, and inside it, coiled up like a rope, was another fish. That second fish, 4.5 times longer than the one that swallowed it. Not a bite, not a piece, the whole thing, intact. What killed this animal wasn't a predator. It wasn't disease. It died because it ate too much. The meal was too big to digest in time. Gas built up inside the body, pushed the stomach outward, and slowly floated the fish up from the deep, mile by mile, until it broke the surface. That's how we found it. And here's the thing that should stop you in your tracks. That's how we find most of them. This fish has never been filmed alive in its natural habitat. Every specimen we've ever studied was already dead. It lives in near total darkness, nearly 2 miles below the surface. We know it exists. We've held it in our hands, but we have never once watched it hunt. We don't even fully agree on how it does it. There are two competing theories. Neither has ever been directly observed, and somehow, despite all of that, this might be one of the most common fish in the entire Atlantic Ocean, an animal that could be everywhere, that we know almost nothing about, whose only real window into our world is the moment everything goes wrong. So what does a creature like this tell us about survival? About what life looks like in a place built entirely around scarcity? Today, we examine the black swallower and what its design reveals about life in an environment where survival depends on taking risks that do not always pay off. But before we do, let me know in the comments, what part of the world are you watching from, and is it day or night there right now? If you enjoy calm videos like this, please give this one a like and maybe subscribe. Now let's continue. At a certain depth, the ocean stops behaving like a place touched by the sun and becomes something else entirely. A layered interior where light is no longer a defining force, but a fading memory that dissolves with each meter downward. Around 200 meters below the surface, the last meaningful traces of daylight lose their structure, thinning into a dim residue that cannot sustain color, cannot carry warmth, and cannot reveal distance. This boundary is not marked by any visible line, yet everything below it exists under a different set of rules. From that point downward, the ocean arranges itself into vast vertical regions. The upper layer, stretching from 200 to 1,000 meters, is known as the twilight zone, a place where light still exists in theory, but not in any form that resembles day. Below it lies the midnight zone, extending from 1,000 to 4,000 meters, where darkness is complete and unbroken. Beneath even that rests the abyss, deeper still, colder, quieter, and further removed from the surface world than any landscape on land. These divisions are not arbitrary classifications, but physical realities that shape every process, every movement, and every living form within them. Within this vertical expanse, the black swallower occupies a narrow band, drifting between roughly 700 and 2,745 meters. At the upper edge of this range, less than 1% of surface light remains, a faint and scattered presence that offers no clarity. By 1,000 meters, even that disappears entirely, leaving an environment where vision no longer serves as a primary sense. Here, darkness is not simply the absence of light, but a stable condition, as constant as gravity. Temperature follows its own quiet discipline. Across this entire range, the water holds steady between 4 and 9 degrees Celsius, just above freezing, with almost no seasonal variation. Unlike the surface, which warms and cools with weather and time, this layer remains unchanged across years, decades, and centuries. It is a global stillness, shared across oceans, uninterrupted by storms or sunlight. Pressure builds in a way that is less visible, but far more absolute. At 1,000 meters, the force pressing inward reaches 100 atmospheres, 100 times the pressure at sea level. Every surface, every structure, every living cell exists under this weight continuously. There is no relief from it, no fluctuation, no moment of release. It defines the limits of what can exist, and it enforces those limits without exception. Yet none of these factors, not the darkness, not the cold, not even the pressure, shapes life here as profoundly as the absence of food. In this part of the ocean, nourishment does not arrive in steady cycles or predictable patterns. It appears rarely, often by chance, drifting down from above or moving through the darkness in brief, fleeting encounters. A single meal may need to sustain an animal for weeks, sometimes longer. Energy is not something to be spent freely. It is something to be guarded, stretched, and extracted with precision. Every organism in this environment reflects that constraint. Movement is measured, bodies are designed for efficiency, and strategies revolve around maximizing the value of each encounter. Nothing here evolves for abundance. Everything evolves for uncertainty. This is not a backdrop or a setting that can be separated from the life within it. It is the force that shapes every structure, every behavior, every decision made without thought. The conditions here are not incidental. They are the reason the black swallower exists in the form it does. After millions of years within total darkness, under constant pressure, in water that barely holds warmth and offers almost nothing to eat, a particular kind of body emerges, one that reflects all of those constraints at once. It looks like this. There is a particular kind of shape that emerges when survival is governed not by speed or strength, but by the careful use of opportunity, a form that appears restrained at first glance, almost modest, until its hidden capacities are revealed. The black swallower, known in scientific terms as Chamazaniger, exists within that quiet category, an animal whose outward simplicity conceals a set of proportions that feel almost misaligned with expectation. Its length rarely exceeds 15 to 20 centimeters, with the largest recorded individuals reaching 25 centimeters, a body no longer than a human hand. At rest, there is nothing immediately dramatic about it. The surface of the body is smooth and uninterrupted, lacking scales entirely, coated instead in a continuous layer of jet black skin that absorbs what little light might ever pass through its world. The color is not decorative. It is a form of absence, a way of becoming indistinguishable from the surrounding water. Seen from above or below, the body narrows into a thin profile, laterally compressed so that its width exceeds its thickness, like a blade turned sideways. This shape reduces resistance as it moves and minimizes the surface it presents to anything that might detect it. It is a form built for subtle passage, for moving through an environment where disturbance carries information. At the front of this narrow body, the structure of the head begins to shift the impression. The lower jaw extends forward beyond the upper, creating a slight overhang that feels deliberate rather than accidental, as if the geometry of the skull has been adjusted to favor a particular kind of motion. The arrangement appears understated until considered in motion, where that forward reach becomes the leading edge of every encounter. The eyes, by contrast, do not follow the pattern seen in many deep-sea species. They remain moderate in size, neither enlarged nor reduced to extremes, a choice that stands apart from the wide, light-gathering eyes common among other inhabitants of similar depths. Their presence suggests that vision, while not absent, is not the primary guide through the surrounding darkness. Other sensory pathways carry more weight, tracing movements that cannot be seen. Within this restrained exterior lies a structure that changes the entire meaning of the animal. When the body is empty, the stomach remains compact, folded inward, and held close, an ordinary organ that gives no indication of what it can become. In preserved specimens that have fed, its transformation is immediate and difficult to reconcile with the size of the fish itself. The stomach extends outward and downward, expanding far beyond the expected boundary of the body, forming a suspended sac that alters the silhouette completely. As it stretches, the tissue thins until it becomes translucent, and in its most extended state, nearly transparent. Through this membrane, the contents inside are visible in detail, not as a vague outline, but as a clearly defined presence, coiled within the body that contains it. The form inside often exceeds the dimensions of the carrier, bending and folding to fit within a space that appears insufficient to hold it. These observations come from specimens that have already completed the process, each one encountered after the fact, offering only a still record of what occurred. The consistency of these forms across multiple examples leaves little ambiguity about the capacity involved. The body accommodates what would otherwise seem impossible, reshaping itself in response to what it has taken in. Nothing in the outer proportions fully prepares for this expansion. The transition from a narrow, unassuming fish to one carrying a volume far exceeding its original outline happens entirely within the same continuous structure. The skin stretches without tearing, the internal space rearranges, and the boundary between inside and outside becomes thin enough to reveal what is held within. The mechanism that allows this begins not in the stomach, but at the point of entry, where the geometry of the head and the arrangement of the jaws define what can be taken in the first place and what happens once that process begins. In most fish, the act of opening the mouth follows a quiet predictability, a rotation around a fixed point, limited by bone and guided by ligament, producing a gape that reflects the size of the head and nothing more. The motion is efficient, repeatable, and bounded, a structure that has remained largely consistent across thousands of species. The black swallower does not follow this pattern. Its jaw is built on a different set of allowances, where the usual constraints have been loosened and extended into something far less intuitive. The lower jaw projects forward beyond the upper, forming a leading edge that initiates contact before the rest of the head arrives. When it opens, it does not stop at the modest angles seen in most fish. It continues upward until the separation approaches 160 degrees, a span that brings the mouth close to a vertical line. At full extension, the opening is no longer defined by the narrow proportions of the skull, but instead expands to match the full width of the body itself, creating a passage that seems disproportionate to the fish that carries it. This expansion is made possible by a structural shift in the upper jaw. In most fish, the bones connecting the jaw to the skull serve primarily as anchors, stabilizing the motion of the lower jaw without contributing significantly to the size of the opening. In the black swallower, this region, known as the suspensorium, becomes an active component of the movement. The bones and cartilage do not simply hold the jaw in place, they allow it to swing outward and downward, increasing the breadth of the gape beyond what the head alone would suggest. The mouth does not just open, it unfolds. Along the edges of this opening runs a single row of sharp teeth on both the upper and lower jaws. The first three on each side are enlarged, forming prominent canines that stand slightly apart from the rest. These teeth are not fixed in a rigid position. They are depressible, able to tilt backward when pressure is applied from the outside, lying flat as something passes inward. Once that pressure is released, they return to their upright position, forming a barrier that resists any movement in the opposite direction. Each tooth sits within a socket that allows a degree of independent motion. They are loose in a controlled way, articulated rather than fixed, capable of shifting to accommodate what enters the mouth. As an object moves inward, the teeth yield, adjusting their angle to reduce resistance. When that same object attempts to move outward, the geometry changes. The teeth align against the direction of movement, creating a surface that grips rather than gives. The difference between entry and exit is built into the structure itself. Additional teeth line the palate, the upper interior of the mouth, forming a second layer that reinforces this directional grip. These are not merely supplementary. They extend the capture mechanism deeper into the oral cavity, ensuring that once something has passed the initial boundary, it encounters further resistance to retreat. In some cases, the length of these teeth is such that those from the upper and lower jaws cross over each other when the mouth closes, interlocking in a way that reduces any open path through which prey might escape. Taken together, these elements form a system that functions less like a simple hinge and more like a controlled passage, one that allows entry with relative ease but resists reversal at every stage. The movement inward is assisted, guided, and accommodated. The movement outward is opposed, redirected, and effectively prevented. This arrangement carries a consequence that extends beyond the moment of capture. Once a prey animal passes into the mouth and the teeth engage, the structure offers no clear mechanism for release. The geometry favors continuation over retreat. The act of seizing becomes a commitment, not through decision, but through design. There is a point at which containment becomes transformation, where an internal space ceases to behave like an organ and begins to function as an extension of the body's outer limits, reshaping the animal from within. In the black swallower, that shift is carried almost entirely by the stomach, a structure that exists in two completely different states depending on whether it is empty or occupied. In its resting condition, the stomach is compact and unremarkable, folded into itself in tight, layered pleats. The tissue gathers inward, forming a dense arrangement that occupies minimal volume, leaving no indication of the capacity it holds. These folds are not random. They are structured in a way that allows them to release gradually, each layer unfolding in sequence as pressure from within begins to build. When prey passes beyond the boundary of the jaws, this unfolding begins. The layers separate, stretch, and extend, expanding outward in a controlled progression that continues far beyond what the external proportions of the fish would suggest. The stomach does not simply enlarge, it reorganizes itself, converting stored surface area into volume. Documented observations show that this expansion can reach up to 10 times the resting capacity, allowing the fish to contain prey whose mass may exceed its own body weight by the same multiple. As the expansion continues, the character of the tissue changes. The walls of the stomach, once thick and folded, become progressively thinner as they are drawn outward. The material stretches to accommodate the shape within, and as it does, it loses opacity. First, it becomes translucent, allowing light to pass through in a diffuse way, and then, at its outermost limit, nearly transparent. The boundary between inside and outside becomes so slight that the form contained within can be seen clearly, its contours pressing against a membrane that was not meant to be visible. The stomach does not remain confined within the original outline of the body. As it fills, it extends beyond it, pushing downward and backward, altering the silhouette entirely. The fish is no longer defined by its initial proportions. It becomes a structure carrying another structure, a thin outer layer stretched around a volume that seems disproportionate to its size. One of the earliest detailed descriptions of this transformation comes from an account written in 1888 by naturalists Kingsley and Hellwald. Observing preserved specimens, they described the expanded stomach as a great bag, projecting both backward and forward, with the fish itself appearing almost displaced above it. The ventral fins, normally positioned along the underside, were described as shifted from their usual placement, drawn away by the expansion. The walls of the stomach and belly had been stretched to such an extent that they were transparent, revealing the identity of the fish contained within. Their words were drawn entirely from still forms, yet the consistency of later findings aligns closely with that early observation. All known examples of this state come from specimens that were no longer alive at the time of discovery. The transformation has never been witnessed in motion, only reconstructed from its outcome. Even so, the physical evidence remains consistent across decades of study, confirming that this is not an anomaly, but a defining feature of the species. Once the stomach reaches this expanded state, the process of digestion begins under conditions that slow it considerably. The surrounding water holds near freezing temperatures, metabolic rates are reduced, and oxygen consumption remains low. Enzymatic activity proceeds at a pace that can extend over weeks, gradually breaking down the contents within, while the stomach maintains its stretched form. This extended digestion introduces a limit, one that is not immediately visible in the structure itself. There is a threshold beyond which the volume and composition of what has been consumed begin to outpace the body's ability to process it. Beyond that point, the balance between containment and breakdown shifts, and the system that allowed the expansion can no longer maintain it. Movement comes first, not as sight or sound, but as a subtle disturbance carried through water, a shifting pattern that travels outward in expanding ripples. In that environment, where light offers no reliable guide, these disturbances become the primary language of presence. The black swallower moves within this field of signals, not scanning with eyes but listening with its entire body, attuned to changes that pass unseen. What is known of its behavior does not come from direct observation. No living individual has been recorded in the act of hunting. Instead, understanding emerges from structure, from preserved forms, and from interpretations first proposed in the 19th century that remain unresolved even now. These ideas are not speculative in the casual sense. They are grounded in anatomy, in the arrangement of tissues and systems that suggest how movement and capture might unfold. There are certain absences that clarify the picture. The black swallower carries no light-producing organs, no filament, no luminous lure suspended in front of its mouth. It does not create its own signal to attract prey. The surrounding darkness remains unbroken by any glow of its making. At the same time, its body is not built for stillness. Descriptions of its musculature and form suggest an animal capable of sustained, directed movement, a swimmer that moves with intent rather than waiting passively for something to drift within reach. The key to this movement lies in a sensory system that runs along its length, a fluid-filled canal embedded within the body, opening to the outside through a series of pores. Each segment carries two of these pressure-sensitive points, and along the head they become more densely arranged, forming a concentrated field of detection. This system, known as the lateral line, registers minute variations in water pressure, tracing the movement of other bodies through the medium itself. It does not see shapes. It perceives patterns, the signature of motion translated into signals that can be followed. Through this system, the fish can detect activity at a distance, distinguishing between the steady drift of the environment and the more defined disturbances produced by living organisms. It navigates by these patterns, adjusting its position in relation to them, closing distance without relying on visual confirmation. The surrounding space becomes a map of movement rather than a landscape of objects. From this foundation, two primary interpretations of its hunting method have been proposed. The first, attributed to the ichthyologist Theodore Gill, suggests an approach from behind. In this model, the fish detects a target through vibration, moves forward, and seizes it by the tail. Once contact is made, the jaws do not simply close and hold. They advance in a coordinated sequence, one side moving forward, then the other, gradually drawing the prey inward in a motion similar to that seen in certain reptiles. The process continues until the entire body of the prey is guided into the stomach, coiling within as it is taken in. The second interpretation proposes a different point of contact. Instead of approaching from behind, the fish engages the head of the prey, biting in a way that restricts movement and respiration. From there, it proceeds to engulf the body progressively, maintaining control as the prey is drawn inward. Both models describe a deliberate, stepwise process rather than a rapid, singular motion. Neither of these sequences has been directly observed. They remain reconstructions, supported by the physical evidence of how prey is found within collected specimens. Despite their differences, both interpretations converge on a shared constraint. Once the initial engagement occurs, once the teeth make contact and begin to function as they are structured to do, reversal becomes unlikely. The mechanism favors continuation, guiding the process forward without offering a clear path back. Within this system lies a limitation that cannot be fully resolved. Detection through vibration provides information about movement and presence, but not always precise scale. A signal that appears within manageable bounds may correspond to something larger than expected. The assessment occurs before contact, but the commitment begins at contact itself, and once that moment passes, the structure carries the process forward without pause. There is a moment, somewhere after the capture is complete, and the body has settled back into the surrounding water, when the outcome is no longer determined by strength or structure, but by time. Inside the expanded form, the process begins quietly, without any visible shift from the outside, a gradual change unfolding within the space that was created to hold what has been taken. The prey, still largely intact, begins to break down through bacterial action. Even in near freezing water, this transformation does not stop. It slows, but it continues, driven by microorganisms already present within the tissues. At the same time, the digestive system of the swallower works in parallel, releasing enzymes, beginning its own slower process of reduction. For smaller meals, the balance holds. Digestion progresses at a pace that keeps ahead of decay, and the contents are gradually reduced and absorbed. When the prey exceeds a certain size, that balance shifts. The rate of decomposition begins to overtake the rate of digestion. The tissues inside the stomach soften, break apart, and release byproducts into the enclosed space. Among these byproducts are gases, primarily carbon dioxide and hydrogen sulfide, generated as the bacterial processes continue. These gases do not dissipate into the surrounding water. They accumulate, held within the stretched boundaries of the stomach. The structure that allowed such expansion offers no pathway for release. The membrane contains everything within it, and as the gases build, the internal volume begins to increase beyond what was created by the prey alone. The stomach, already extended close to its physical limits, is forced outward further, its surface drawn thinner still as pressure builds from within. With that accumulation comes a shift in buoyancy. The added gas reduces the overall density of the fish, altering its relationship with the surrounding water. What once held position within a specific depth range begins to drift upward. The movement is slow at first, a gradual rise that requires no active motion, driven entirely by the difference between the body and the water around it. As the fish ascends, the surrounding pressure decreases. Each meter upward reduces the force pressing inward, and with that reduction, the gases inside expand. The volume increases further, amplifying the buoyant effect. This creates a cycle that reinforces itself, expansion leading to ascent, ascent leading to further expansion. The process does not stabilize. It accelerates. The fish cannot counter this movement. There is no mechanism within its body to expel the gas, no adjustment that can restore its previous density. Swimming downward would require overcoming both buoyancy and the physical burden of the expanded stomach, a condition the body is not structured to manage. The path becomes one-directional, dictated by physics rather than behavior. It passes upward through layers of water that differ from the ones it was adapted to inhabit. Pressure continues to drop. Temperature begins to rise slightly, and faint traces of light reappear, increasing with each meter. These changes occur gradually in space, but they represent a rapid departure from the stable conditions that shaped the animal's biology. Eventually, the ascent brings it to the surface. There, it is no longer supported in the same way. The body, already under internal strain, cannot maintain its structure. It is found floating, the stomach distended far beyond its resting form, sometimes ruptured, sometimes intact but stretched to an extreme. Within it, the remains of the prey are still visible, preserved in the position they occupied when the process began. Most of what is known about this species has come from individuals encountered in this state. They appear not as representatives of successful feeding, but as records of a specific failure, each one following the same sequence, each one ending at the same boundary between depth and air. And yet the species remains present across vast regions of the ocean. The pattern continues because, in most cases, the balance holds long enough for digestion to complete. Those that succeed do not rise. They remain below, completing the process unseen, their bodies returning to their original form as the contents are reduced. By the time this particular individual reached the surface, the sequence that brought it there had already completed itself in full, leaving behind a form that could be measured, examined, and recorded with unusual clarity. Among all known cases, this one remains the most thoroughly documented, not because it was unique in mechanism, but because of how precisely its details were preserved. The fish itself measured 19 centimeters in length, roughly 7.5 inches from head to tail, a size consistent with the upper range of its species. Within its distended body was another fish, a snake mackerel identified as Gempelus serpens, measuring 86 centimeters or approximately 34 inches. The proportions are difficult to reconcile at first glance. The prey was 4.5 times longer than the predator, a length difference that exceeded not just typical feeding limits, but the apparent physical scale of the swallower itself. The specimen was recovered off the southern coast of Grand Cayman Island in 2007, not caught deliberately, but encountered drifting at the surface. A fisherman brought it aboard, and its unusual form drew immediate attention. A marine biologist present at the time documented the specimen through photographs, capturing the condition in which it was found. These images were later forwarded to the Harbor Branch Oceanographic Institute in Florida, where the species identification was confirmed and the case entered into the scientific record. Within those images, the stomach appears fully expanded, its walls stretched thin enough to reveal the entire outline of the prey inside. The mackerel is coiled within the transparent boundary, its body bent to fit the available space, its structure still clearly defined. The swallower itself is no longer recognizable as a compact fish. It becomes a framework surrounding a larger form, its own proportions altered beyond their resting state. The presence of the snake mackerel in this context offers a clue to where the encounter likely occurred. This species typically inhabits shallower and warmer waters than those occupied by the black swallower during daylight hours. The overlap between them would not occur in the deeper range, but rather during the vertical movements that take place under cover of darkness. As the swallower ascends at night in search of food, it enters regions where such prey are more commonly found. At some point during this upward movement, the two paths intersected. The lateral signals would have been sufficient to indicate motion and presence, but not necessarily proportion. The prey, by any standard, exceeded the manageable range. Yet once contact was made, the structure of the jaw ensured that the process continued. The teeth engaged, the initial grip was established, and the sequence that follows from that engagement began to unfold. From there, the outcome was governed by the same constraints seen in other cases, but amplified by the scale involved. The stomach expanded to accommodate the length of the mackerel, its internal space reorganized to contain what had been taken in. Digestion began, but the size of the prey placed it beyond the threshold where processing could keep pace with decomposition. The internal changes followed accordingly, leading to the rise that brought the specimen into view. What distinguishes this case is not only the magnitude of the difference between predator and prey, but the clarity with which each stage can be inferred from the final state. The measurements align with the known limits of expansion. The position of the prey reflects the inward progression described in anatomical studies, and the condition of the stomach corresponds to the process that leads to ascent. The sequence does not depend on chance in the way it unfolds once initiated. It follows directly from the structure of the body itself. The engagement of the teeth sets a path that continues without interruption, and the anatomy does not provide a mechanism to alter that path once it has begun. In this instance, the scale of the prey ensured that the outcome would extend beyond what the system could sustain, but the progression toward that point remained consistent with the same design that allows the species to persist across its range. Long before the surface settles into darkness, a shift begins below, subtle at first, then gathering into a coordinated movement that spans entire ocean basins. It is not marked by sound or light, only by direction, a collective rising that repeats with such regularity that it becomes part of the planet's rhythm. Across every ocean, from equatorial waters to colder regions, countless organisms begin to move upward together. This movement includes fish, squid, shrimp, and zooplankton, each following the same vertical path, ascending hundreds of meters through layers that change gradually in pressure and temperature. They rise under the cover of night, when the reduced visibility above lowers the risk of detection from predators that depend on sight. The ascent is not hurried. It unfolds steadily, carried by a balance between caution and necessity, as these organisms approach regions where food is more abundant. This daily movement is known as diel, vertical migration, and in terms of total biomass, it is the largest recurring movement of life on Earth. Each night, more living mass shifts upward and then downward again than in any migration observed on land. It is continuous, global, and largely unseen, taking place in darkness across distances that extend far beyond the scale of individual perception. Within this vast flow, the black swallower is almost certainly present, moving along the same vertical pathway. During daylight hours, it remains within deeper ranges, between 700 and 2,745 meters, where conditions remain stable. As darkness settles above, it begins to ascend, entering layers where the density of potential prey increases significantly. The transition is gradual, but the difference in available food becomes pronounced as it approaches shallower depths. As it rises, the surrounding environment shifts in ways that subtly influence its movement. Pressure decreases incrementally, reducing the compression that defines deeper regions. Temperature rises slightly, introducing a faint gradient that contrasts with the uniform cold below. At the same time, the number of moving organisms increases, filling the water with overlapping signals of motion. For a species that detects its surroundings through pressure changes, this creates a more active and information-rich field. It is within these upper layers that encounters become more likely. The snake mackerel from the Grand Cayman specimen occupied this range, moving within waters that the black swallower would only reach during its nighttime ascent. The convergence of these two species would not occur in the deeper zones, but within this shared vertical space, where prey becomes more accessible and interactions more frequent. Many of the recorded cases of overextended feeding appear to originate from this region, where the abundance of potential targets increases both opportunity and risk. As the night progresses, the upward movement slows and eventually stabilizes. Then, as the first traces of daylight begin to return to the surface, the direction reverses. The same organisms that rose begin to descend, retracing their path downward, returning to the depths where they spend the daylight hours. This reversal is as consistent as the ascent, part of a cycle that repeats without interruption. The black swallower follows this pattern, carrying whatever it has taken during the night back into deeper water. The descent brings it again into the colder, more stable environment where digestion proceeds slowly. A large meal, once secured, may remain within the body for weeks, processed gradually as the fish maintains its position within these lower layers. This vertical journey occurs every day, extending across thousands of meters, upward and downward, without reliance on external markers or visual cues. The movement is guided by internal responses to pressure, to chemical gradients, and to the shifting distribution of other organisms. It is a form of navigation embedded within the body itself. Within this immense and continuous migration, the black swallower moves as a single, small participant, its length no greater than a ruler, traveling through one of the largest coordinated movements of life on the planet, carried by patterns that have repeated for longer than any record of observation, seen again in stillness. After the movements and thresholds that define its life, the body resolves into a set of quiet decisions, each one shaped by the conditions it passes through. Nothing on it announces itself loudly. The form is spare, composed of surfaces and edges that seem almost understated until considered in relation to what surrounds it. The skin carries no scales. It lies smooth along the length of the body, uninterrupted from head to tail, reducing friction as water moves past. This surface does more than ease motion. It minimizes the disturbances that travel outward when the fish shifts position, keeping pressure waves small and difficult to distinguish from the background. In an environment where detection can occur through subtle changes in flow, this reduction becomes part of concealment. The same surface also limits reflection. In the rare moments when brief flashes of bioluminescence pass nearby, there is little for that light to catch, little to return to the surrounding space. The color deepens that effect. The body is not merely dark but fully black, absorbing what little illumination might reach it. Below 200 meters, where the only light comes in short, chemical bursts. This coloration prevents the body from outlining itself against those flashes. Instead of appearing as a defined object, it blends into the absence around it, becoming less a figure within the water and more a gap within it. The eyes, moderate in size, sit within this dark surface without exaggeration. They do not extend outward or enlarge to gather light, a departure from the patterns seen in many other species that share similar depths. Their presence remains functional but secondary, contributing to awareness without dominating it. The primary stream of information arrives through the lateral line, a continuous channel running along the body, punctuated by pores that register minute shifts in pressure. Along the head, these openings become more numerous, forming a concentrated array that refines the resolution of this sense. Through this system, motion is perceived not as an image, but as a pattern moving through the surrounding medium. This difference in perception shapes the role the body plays in movement. It does not orient itself toward visible targets, but adjusts in response to gradients and signals that pass across its surface. The distinction between predator and environment becomes less about what is seen and more about what is felt, the body acting as an instrument that reads changes in the water itself. The form of the body supports this approach. It remains laterally compressed, narrowing when viewed from above or below, presenting a thin profile that reduces its visibility against faint downwelling light and against upward flashes from organisms below. This shape also allows for controlled, precise movement, reducing resistance in directions that matter most during approach. Along the underside, just forward of the gills, a single spine extends, small but distinct. Its purpose is not fully understood, yet it remains consistent across specimens, suggesting a role that has not been fully resolved through observation. It sits there as part of the structure, neither prominent nor absent, contributing to the overall design without drawing attention to itself. Two dorsal fins rise along the back. The first carries between 10 and 12 spines, rigid elements that provide stability during movement. Behind it, the second fin extends longer, composed of softer rays that allow for subtle adjustments. Together, they give the fish a degree of maneuverability that allows it to change direction with precision, to correct its path in response to shifting signals, and to maintain control within a medium that offers no fixed reference points. Taken as a whole, the body does not appear assembled through excess or ornament. Each feature aligns with a function, each omission with an advantage gained through absence. The smooth surface, the black coloration, the measured eyes, the sensory line, the compressed form, the fins, and even the small, unresolved spine all contribute to a structure that operates within its environment with minimal waste. It is not a collection of traits placed side by side, but a continuous solution shaped by the same forces that define the space it moves through. There is a small, mechanical detail hidden within the mouth that alters the meaning of everything that comes before it, a feature so subtle in appearance that it can be overlooked, yet so decisive in consequence that it reshapes the entire sequence of events. It is not found in the size of the jaws or the reach of the gape, but in the way the teeth themselves respond to motion. Each tooth is set at a backward angle, oriented toward the throat rather than outward. When something presses inward, the teeth yield. They fold slightly along their base, lowering resistance and allowing passage deeper into the mouth. This movement is not passive in the sense of collapse. It is controlled, built into the structure, a directional flexibility that responds to pressure from a specific side. When the direction of force changes, the behavior reverses. The same teeth that bent inward return to their upright position, restoring their angle and creating a surface that resists outward movement. In this orientation, they act less like simple points and more like a series of hooks, each one aligned to catch against anything that attempts to retreat. The difference between entry and exit is defined entirely by this shift, a transition that occurs automatically, without adjustment or delay. Across multiple examinations, this arrangement has led to a consistent interpretation. The black swallower likely lacks the ability to release prey once it has passed beyond a certain point. The structure does not provide a mechanism for reversal. There is no coordinated action that can reorient the teeth in a way that would allow something to move outward freely. The design favors continuation, guiding movement deeper rather than permitting it to return. Within this framework, the act of feeding takes on a different character. It is not a sequence that can be paused or reconsidered once it begins. The initial contact, the moment when the teeth engage and respond to pressure, establishes a direction that the body then follows. The process that draws prey inward is supported at every stage by the arrangement of the teeth, each segment reinforcing the next, creating a progression that moves in a single direction. This removes a degree of control that might otherwise be assumed. The fish does not assess, grasp, and then decide whether to proceed. The structure of its mouth bypasses that sequence. Once engagement occurs, the continuation is not optional. The anatomy itself carries the process forward, independent of any capacity to halt or reverse it. Seen in this way, the events that lead to overextension are not the result of excess or error in the usual sense. The same mechanism that allows the fish to secure prey in an environment where opportunities are limited also prevents it from disengaging when those opportunities exceed manageable bounds. The feature that ensures success under scarcity also sets the conditions for failure when scale exceeds capacity. In an environment where encounters are infrequent, hesitation carries its own cost. A prey item that passes within reach may not appear again for an extended period. A structure that commits fully to each opportunity increases the likelihood of securing food when it is available. The teeth, in their willingness to engage anything that enters their range, reflect this pressure. They do not discriminate based on size at the moment of contact. They respond to presence. This characteristic cannot be separated into advantage and disadvantage. It exists as both simultaneously. A single design that produces two different outcomes depending on the circumstances of each encounter. The strength of the system lies in its ability to secure what would otherwise be lost. The vulnerability arises from that same inability to release what has been secured. In this balance, the boundary between success and failure is defined not by intention, but by scale, and the mechanism that governs both remains unchanged across every instance. What passes through the body leaves a record, a quiet archive preserved within tissue that no longer moves, where each meal becomes a fragment of evidence. From these fragments, patterns begin to form, not through observation of behavior, but through accumulation, each specimen adding a small piece to a larger picture. In one of the most comprehensive examinations to date, 337 individuals of the genus Chiasmodon were studied from the Gulf of Mexico, collected between 2010 and 2011. Within them, the contents of the stomach offered a consistent outline of what this animal takes from its environment. Cephalopods appear most frequently, present in 39% of examined stomachs. These include squid and related forms, soft-bodied organisms that move through the same depths, leaving behind the kinds of signals that the swallower can detect. Close behind, teleost fish, or bony fish, account for 35%. forming nearly an equal portion of the diet. These are not random captures but recurring presences, species that occupy similar layers of the ocean and intersect with the swallower's movements. Crustaceans, despite their abundance in these waters, appear in only 2% of cases. Their relative absence suggests a form of selection, not necessarily through preference in the conscious sense, but through compatibility with the mechanisms of capture and ingestion. Their harder exoskeletons and smaller size may place them outside the range of what the swallower's feeding strategy is optimized to handle, even though they are readily available. Among the identifiable prey are lanternfish, snake mackerels, deep-sea eels, and a range of other mesopelagic fish. Each of these species carries its own pattern of movement, its own presence within the water column, contributing to the signals that guide the swallower's approach. They are not chosen in isolation. They are encountered within the same shifting field, drawn from a shared environment where paths cross without warning. When these observations are translated into energy intake, another pattern emerges. The estimated daily ration for the black swallower averages 3.64% of its body weight. This value stands above the range observed in most other mesopelagic fish, which typically fall between 1.5 and 2.7%. The difference reflects not a constant intake, but a strategy built around larger, less frequent meals. The swallower takes in more energy at once, then relies on that intake over extended periods. This approach places it at a different point along the spectrum of feeding strategies. Rather than sustaining itself through continuous small captures, it depends on intermittent, substantial intake. The risk associated with each event increases, but so does the potential return. Each successful capture carries enough energy to sustain the body through long intervals, aligning with the slow processes that govern digestion in its environment. Within the broader community of deep-sea predators, this pattern reduces direct overlap. Other species occupying similar depths often focus on smaller, more numerous prey. The dragonfish, belonging to the family Stomiidae, for example, frequently target lanternfish as a primary resource. While lanternfish also appear in the swallower's diet, they do not define it. The overlap remains partial, leaving space for each to operate without constant competition. This distribution of prey among different predators forms a structure known as niche partitioning. It is not an arrangement imposed from outside, but one that emerges as species adapt to specific roles within the same environment. By focusing on different types or sizes of prey, each predator reduces direct competition, allowing multiple strategies to coexist within the same depth range. The black swallower occupies a position within this structure that is defined by scale. Its feeding mechanism allows it to take in prey that exceed the limits of most other fish at similar depths. This capacity places it in a niche where few others can follow, not because the prey are rare, but because the ability to process them is uncommon. In this way, its diet is shaped as much by what it can take as by what is available. The prey it consumes are not exclusive to its environment, but the manner in which it consumes them sets it apart, creating a role that aligns with the structure of its body and the constraints of the world it inhabits. Pressure does not arrive as a sudden force in this part of the ocean. It accumulates gradually, layer by layer, increasing with depth until it becomes the dominant condition, shaping everything within it. At 700 meters, near the upper boundary of the black swallower's range, the surrounding water presses inward at approximately 70 atmospheres. With each descent, that force rises steadily, reaching beyond 270 atmospheres at 2,745 meters. the deepest confirmed record for this species. There is no fluctuation, no relief, only a continuous, unchanging compression that defines the space it inhabits. To place this in another frame, a human body begins to experience physiological stress at around 3 atmospheres. At 10 atmospheres, unprotected exposure becomes rapidly fatal. At the depths where the black swallower moves, the pressure exceeds levels that would compress human tissue to a fraction of its original volume, collapsing air spaces and disrupting every system that depends on them. The contrast is not simply one of tolerance, but of fundamental structure. The bodies of deep-sea fish are built without reliance on compressible gases. They lack conventional air-filled cavities such as those found in surface-dwelling species. The swim bladder, when present, is either reduced or modified, often containing dense fluid rather than gas, eliminating the vulnerability that comes from compression. Without internal spaces that can collapse, the body remains stable as pressure increases, maintaining its shape even as the surrounding force intensifies. At the level of cells, further adjustments support this stability. The membranes that enclose each cell are composed in a way that preserves their flexibility under compression. They contain a higher proportion of unsaturated fatty acids, which prevent the membrane from becoming rigid when pressure rises. This fluidity allows cellular processes to continue, ensuring that exchange and movement across these boundaries remain possible even under extreme conditions. Proteins, the structures responsible for most biological functions, are also configured differently. Their shapes, defined by the way they fold, are stabilized by the surrounding pressure. In environments closer to the surface, where pressure is lower, these same configurations become unstable. The folding that supports function at depth depends on the force pressing inward, and without it, the structures lose their integrity. Enzymatic activity, structural support, and metabolic processes all rely on this precise arrangement. When deep-sea fish are brought upward rapidly, this balance is disrupted. As pressure decreases, the conditions that maintained the structure of proteins and membranes change. The internal systems begin to fail, not through visible damage alone, but through the breakdown of the biochemical processes that sustain life. The body may remain intact for a short time, but the functions that define living tissue can no longer operate as they did at depth. For the black swallower, this relationship with pressure is not a matter of tolerance, but of dependence. Its entire physiology is calibrated to function within a narrow range of conditions, defined by depth. The pressure is not an external challenge to be endured. It is part of the environment that allows its internal systems to exist in a stable state. Remove that pressure, and the balance is lost. The structures that enable movement, digestion, and perception cease to operate as intended. The body, which is fully functional under compression, cannot maintain those functions in its absence. This dependence creates a boundary that is not marked by distance or temperature, but by physics itself. The region it occupies remains separate from the surface world, not by isolation in space, but by the conditions that define it. The two environments exist within the same ocean, yet they are governed by different rules, and the transition between them carries consequences that cannot be reversed. No living black swallower has been observed crossing that boundary and continuing to function. The separation holds, maintained by the same forces that shape its existence below. There is an absence that sits quietly at the center of everything known about this animal. Not a gap in data that might eventually be filled, but a continuous condition that has persisted for as long as it has been studied. Across decades of research, across expeditions that have reached into nearly every accessible layer of the ocean, one detail remains unchanged. The black swallower has never been seen alive in its natural environment. This absence stands in contrast to the tools that have been used to explore the same depths. Over the past 30 years, remotely operated vehicles have moved through the mesopelagic and upper bathypelagic zones with increasing precision. They descend with powerful lights, with high-resolution cameras, with instruments capable of recording fine detail across extended periods. These systems have documented thousands of species, revealing behaviors, interactions, and forms that were once entirely unknown. Within that growing archive, the black swallower does not appear. It is not present in the recordings, not in passing frames, not in distant silhouettes. The absence is consistent across different regions, across different deployments, across different technological approaches. It remains outside the range of what has been captured. This absence does not reflect rarity. In scientific literature, the species is described as widespread and common, particularly in the North Atlantic, where it is considered the most abundant member of its genus. Its distribution extends across multiple oceans, with confirmed records reaching depths of 3,900 meters and shallower occurrences around 150 meters in juvenile stages. It occupies a range that overlaps with many areas explored by submersible systems, yet it remains unrecorded within them. Several possibilities exist to account for this. The fish may respond to the presence of artificial light, moving away from the illumination before it can be documented. It may inhabit regions of the water column that are less frequently targeted, remaining within layers that are difficult to sample consistently. It may move in ways that reduce the likelihood of encounter, passing through spaces that are vast relative to the limited field of view of any instrument. Each of these explanations carries some plausibility, yet none has been confirmed. All direct physical evidence of the species comes from individuals that are no longer alive at the time of observation. Specimens are found floating at the surface after internal processes have altered their buoyancy, or they are recovered through trawl operations that bring them upward from depth. In both cases, the condition of the animal reflects a state after death, shaped either by the events that led it to the surface or by the pressure changes experienced during collection. Every photograph, every preserved specimen, every anatomical description is based on these remains. The body can be measured, the structure can be examined, the contents can be analyzed, yet the living form, the movement, the behavior within its environment remain unobserved. The sequence of actions that define its existence has been inferred, reconstructed from evidence that arrives only at the end of the process. This situation has remained unchanged for nearly 200 years. Since its first recorded collection in the early 19th century, the species has been studied through the same indirect means. Advances in technology have expanded the range of observation for many other deep-sea organisms, yet this particular animal continues to exist outside that expanding boundary. Within this context, the black swallower occupies a peculiar position. It is not hidden in the sense of being inaccessible, nor is it unknown in terms of distribution or structure. It is present, documented in form and frequency, yet absent in life. The space it occupies overlaps with areas that have been explored extensively, yet it remains beyond direct observation. The result is a portrait constructed entirely from stillness, an understanding built from what remains after processes have concluded. The living sequence, the continuous motion within its environment, has not been seen, leaving a space where behavior is understood through inference rather than witness, sustained by evidence that arrives only after the fact, long before cameras descended into the ocean and long before instruments could translate pressure into numbers. The first encounter with this animal arrived in a far simpler way, lifted from the depths on a line, its form already still, its life already concluded. In 1830, off the coast of Madeira in Portugal, a naturalist named Lowe retrieved a specimen from approximately 312 fathoms, or around 570 meters below the surface. The fish was brought upward, placed among collected materials, and then, for a time, it remained without description, a presence in a collection that had yet to be fully understood. For 12 years, it stayed that way, preserved but unnamed, its structure available but its significance not yet recognized. It was only in 1842 that the species was revisited at the same location, again off Madeira, when Johnson examined it more closely and began the process of formal description. The details that had remained unarticulated were then recorded, and by 1864, the species was given its scientific name, Chiasmodon niger. The name itself reflects what could be observed from the body alone. Chiasmodon draws from the Greek words chiasma, meaning cross, and odon, meaning tooth, a reference to the distinctive arrangement of the teeth within the mouth. Niger, from Latin, denotes black, capturing the coloration that defines its exterior. Together, the name describes features that were visible, measurable, and consistent across specimens, even in the absence of any observation of movement. These early descriptions were based entirely on individuals that had already died. The scientists who named and classified the species had no opportunity to observe it in motion, no way to witness how it fed, moved, or interacted with its environment. The entire understanding of its form emerged from stillness, from bodies recovered after processes had completed, offering only the final state of structures that had once been active. By 1888, further accounts began to expand on what could be inferred from these preserved specimens. In the Riverside Natural History, naturalists Kingsley and Hellwald described the expanded stomach in detail, noting how it extended far beyond the expected boundaries of the body. They wrote of a great bag, projecting both backward and forward, of the ventral fins displaced from their usual position, and of the walls of the stomach stretched to such a degree that they became transparent, revealing the form of the prey contained within. Their description, derived entirely from observation of dead specimens, remains closely aligned with what later examinations have confirmed. A few years later, in 1904, the sailor and naturalist Frank Thomas Bullen offered his own account in Creatures of the Sea. He described the fish as a veritable nightmare in appearance, noting that its mouth seemed to cleave the head laterally for its whole length, leaving it without a visible profile when fully open. His language reflects an attempt to convey the unfamiliar proportions of the animal, translating structure into imagery drawn from human perception. These early observers worked within the limits of what they could access, relying on physical forms that had already completed their function. Their interpretations were shaped by what remained, yet they captured key aspects of the animal with a level of accuracy that continues to hold. Without direct observation, they assembled an understanding that aligned closely with later findings, guided by careful attention to detail. The lineage to which this species belongs extends far beyond these first encounters. Fossil evidence from the Middle Miocene, including the genus Banicovictus found in Italy, places the origins of this family at approximately 13 million years ago. These fossils represent more basal forms, indicating that the structural characteristics seen today have been part of this lineage for millions of years. During that span, the deep ocean has remained a stable environment, and the forms adapted to it have persisted with relatively little change. The individuals examined by Victorian naturalists were part of a continuum that extends far into the past, their bodies shaped by processes that had already been in place long before human observation began. The early descriptions, drawn from still forms lifted from the water, capture a moment where human understanding first intersected with a structure refined over immense stretches of time, revealing a design that had been operating unseen beneath the surface for millions of years. Before the body settles into the depths that define its later life, it begins in a place that bears little resemblance to that final environment, carried within water that still holds light, warmth, and a different rhythm of movement. The earliest stage exists not in darkness, but within the open layers nearer the surface, where the processes of reproduction unfold without the constraints that shape adulthood. The species is oviparous, releasing eggs directly into the surrounding water. Each egg measures between 1.1 and 1.3 millimeters in diameter, smaller than a sesame seed, a nearly weightless sphere drifting within the currents. At its center lies a clear oil globule, providing buoyancy and energy, while 6 dark pigment patches form along its structure. These patches do not remain fixed. As development proceeds, they shift position, migrating along the length of the emerging larva, aligning themselves from just in front of the developing eyes to the tip of the embryonic spine. Observations of these eggs have been most frequent in winter, particularly off the coast of South Africa, suggesting a seasonal pattern to spawning, yet the early stages do not remain confined to that region. Juvenile individuals have been recorded from April to August near Bermuda, indicating that the early life phases span large distances, carried by currents across thousands of kilometers between spawning and growth. In these early stages, the form differs markedly from the adult. The juvenile body is covered in small projecting spinules, fine extensions along the skin that create a textured surface. These structures are temporary, present only during this phase, and they gradually disappear as the organism matures. Their exact function is not fully resolved, but their consistent presence suggests a role suited to life near the surface, where conditions differ from those deeper below. The youngest individuals occupy relatively shallow water, often close to the surface itself, drifting among plankton in regions where sunlight penetrates and supports a broader range of life. Here, the environment is more dynamic, influenced by currents, temperature variations, and the presence of other organisms that move within this illuminated space. The juveniles exist within this layer, not yet shaped by the conditions that will define their later form. As growth continues, a gradual shift begins. The movement downward does not occur as a single transition, but as a continuous descent, with individuals occupying progressively deeper layers over time. The range extends downward to approximately 1,050 meters during this stage, bridging the boundary between light and darkness. The change is not marked by a clear dividing line, but by a gradual reduction in light, temperature, and the availability of surface-driven influences. During this descent, the body begins to change. The spinules recede, leaving the skin smooth. The coloration deepens, moving toward the uniform black seen in adults. The proportions adjust, aligning with the structure that will later support the feeding strategy and movement within deeper water. These changes occur incrementally, each stage reflecting the conditions of the depth it occupies. At some point along this downward path, the juvenile crosses into a region where light no longer reaches in any meaningful form. The exact depth at which this occurs is not precisely defined, and no direct observation has captured the moment. The transition from a world shaped by sunlight to one defined by its absence remains unrecorded, inferred only from the distribution of specimens across different depths. No individual has been followed continuously from this early stage to adulthood. The connection between the surface-dwelling larva and the deep-dwelling adult is established through collected specimens at different points along this path, each representing a stage within the sequence. The full journey, from egg to mature form, exists as a series of separate observations rather than a single continuous record. The life cycle begins within illuminated water, shaped by conditions that differ entirely from those found below. Over time, it moves downward, leaving that environment behind, entering regions where the defining features of the surface no longer apply. The progression continues until the body reaches a depth where light has no presence, completing a transition that spans the full vertical extent of the ocean's inhabited layers, a movement from one world into another that remains unwitnessed in its entirety. Time stretches differently once the intake has been secured and the body settles back into its deeper range, where motion gives way to a slower internal sequence that unfolds beyond immediate perception. The initial act of feeding, abrupt and decisive, gives way to a prolonged interval in which very little appears to change from the outside, yet a sustained process continues within. The surrounding water holds steady within a narrow thermal band between 4.2 and 8.7 degrees Celsius, with an average near 4.9 degrees Celsius, a range established across hundreds of observations. At these temperatures, metabolic activity slows considerably. Enzymatic reactions that proceed rapidly in warmer conditions operate here at a reduced pace, extending the time required for each stage of digestion. The chemical processes that break down a large meal do not stop, but they proceed gradually, unfolding over an extended period that may last for weeks, and in some cases, longer. During this interval, the body remains largely still. Movement is minimized, not as a passive state, but as a means of preserving the energy gained from the meal. The fish maintains its position within the water column, adjusting only as needed to remain within its preferred depth range. The vertical migrations continue, rising and descending in accordance with the broader patterns of the environment, yet, outside of this cycle, activity is reduced to what is necessary. The stomach, once expanded, begins a gradual contraction. The outer boundary, previously stretched thin, recedes as the contents within are processed and reduced. The volume decreases incrementally, returning toward its resting form. This change is not abrupt. It follows the pace of digestion, reflecting the steady conversion of the ingested material into energy and waste. The structure that once extended beyond the body's outline draws inward, restoring the original proportions over time. Throughout this phase, the body does not engage in further feeding. The space required for digestion is fully occupied, and the processes underway limit the capacity to take in additional material. The structure that enabled the initial intake now defines a period of exclusivity, during which the fish remains committed to processing what it has already secured. As the internal work progresses, the stored energy is distributed, supporting the body through a period in which no new intake occurs. The balance between consumption and expenditure is maintained over this extended interval, allowing the fish to persist without the need for continuous feeding. The pacing of this cycle aligns with the conditions of the environment, where opportunities for feeding are not frequent or predictable. Eventually, the digestion reaches completion. The remaining material is reduced, the stomach returns fully to its compact state, and the body resumes its original silhouette. At this point, the stored energy has been converted, and the reserves begin to decline. The period of inactivity gives way to renewed readiness, not through a sudden shift, but through a gradual return to the state in which detection becomes primary once again. The lateral line, which had remained responsive throughout, now becomes the central guide once more, attuned to the movements that pass through the surrounding water. The body transitions from processing to anticipation, moving from internal activity to external awareness. This sequence, from intake to extended digestion and back to detection, forms a continuous cycle that aligns with the constraints of the environment. The intervals between feeding are not uniform, and the size of each meal can vary significantly. The body is structured to accommodate this variability, absorbing large amounts of energy when available and sustaining itself through prolonged periods without intake. Within this pattern, there is no steady middle state. The rhythm alternates between the presence of a substantial meal and the absence of any intake, each phase defined by the same underlying conditions. The transitions between them are gradual, shaped by temperature, pressure, and the availability of food, forming a cycle that repeats without fixed timing, guided by the environment rather than by any regular schedule. Classification often moves more slowly than discovery, arranging organisms into structures that reflect what can be seen at the time, then shifting as new methods reveal connections that were previously hidden. The black swallower occupies one such shifting position, placed within a lineage that extends across multiple oceans, yet tied to relatives that do not immediately resemble it in form or behavior. It belongs to the family Chiasmodontidae, a group commonly referred to as the snake-toothed fishes or swallowers. Within this family are 4 genera and 32 recognized species, all predatory, all adapted to life in deep water, and all distributed across the major ocean basins. Despite variations in size and detail, they share a set of structural traits that align them, particularly in the arrangement of the jaws and teeth, and in the capacity to take in prey that challenges the apparent limits of their bodies. For much of the 20th century, this family was placed within the order Perciformes, a broad classification that grouped together many perch-like fishes based on outward similarities in form. This arrangement reflected the best available understanding at the time, relying on visible structures such as fin placement, body shape, and skeletal features to establish relationships between species. In 2017, a different approach reshaped this placement. Using molecular and genomic analysis across nearly 2,000 fish species, researchers re-examined these relationships at a deeper level, comparing sequences of genetic material rather than external characteristics alone. This analysis led to the reclassification of the Chiasmodontidae into the order Scombriformes, a group that includes some of the most recognizable and powerful predators in the ocean. Within this order are tunas, mackerels, swordfish, and billfishes, species known for speed, strength, and life near the surface. The connection places the black swallower within the same broader lineage as these animals, despite the clear differences in appearance and habitat. The contrast is immediate when considered in terms of behavior. A bluefin tuna, moving through surface waters, can reach speeds exceeding 70 kilometers per hour, covering large distances in short bursts as it pursues prey. The black swallower, by comparison, occupies a space where movement is measured, where encounters are infrequent, and where the act of feeding unfolds over extended periods. The link between them is not found in how they move, but in the underlying strategy that shapes their role as predators. Both are structured around the idea of high return from each successful capture. For the tuna, this return is realized through rapid pursuit and immediate consumption. For the swallower, it emerges through the ability to secure and process a large meal that sustains the body over time. The expression differs, but the principle aligns. Within its own genus, Chiasmodon, there are 7 additional species, Each occupies a similar ecological space, sharing the defining characteristics of the family while differing in distribution and specific traits. Among them, the black swallower stands as the most widespread and the most frequently encountered in scientific records, particularly within the North Atlantic. It serves as the primary reference point for understanding the group, not because it is fundamentally different, but because it is the one most often available for study. Even with this relative availability, the level of understanding remains limited. The family as a whole is defined more by structure than by observed behavior, and the connections between species are drawn from anatomy and genetic data rather than direct observation of how they live. The others within the group remain less documented, their presence known through specimens, but their lives largely unrecorded. In this arrangement, the black swallower sits within a lineage that bridges extremes, linking the slow processes of the deep with the rapid movements of the surface, connected through evolutionary pathways that extend beyond immediate appearance, forming relationships that become visible only when examined at a deeper level. Maps suggest boundaries, lines that divide one region from another, names that fix location into something stable and known. The black swallower does not follow those lines. Its presence ignores borders, currents of politics and geography, passing far above a layer of water that remains consistent in ways the surface never is. It appears in records from the Atlantic, the Pacific, and the Indian Ocean, extending through waters surrounding South Africa, Australia, New Zealand, the Galapagos, Iceland, Bermuda, Brazil, Japan, and more than 100 additional national territories. These entries accumulate across decades, each specimen a point on a chart, each recovery reinforcing the same pattern. The fish is not confined to a coastline, not tied to a basin, not restricted to a single hemisphere. It is, in the most literal sense, global. Yet the pattern resolves differently when viewed beyond the surface. The distribution does not spread evenly through the water column, nor does it align with the shapes of continents. Instead, it gathers within a narrow range that remains consistent regardless of where it is found. Temperature defines that range, holding steady between 4.2 and 8.7 degrees Celsius, with a mean near 4.9, a band of cold that threads its way through the oceans at depth. This band does not sit at a fixed distance from the surface. It rises and falls depending on latitude, ocean structure, and seasonal variation, but it persists as a continuous layer, wrapping around the planet beneath the shifting surface waters. In tropical regions, it lies deeper, descending toward 2,745 meters, while in cooler regions, it may approach shallower limits closer to 700 meters. The exact depth changes, but the condition remains. Within this layer, pressure and darkness align with temperature, forming a consistent environment that exists independently of geography. The water is dense, lightless, and near freezing, a stable domain compared to the variability above. It is here that the black swallower remains, not anchored to place, but to a set of physical parameters that define where it can function. This creates a different kind of distribution, one that does not map cleanly onto the world as it is usually understood. A single individual may move vertically through this band, following nightly changes, yet always returning to the same thermal range. Across oceans, others do the same, each occupying a segment of a continuous layer that spans thousands of kilometers. The species becomes less a collection of populations in separate locations and more a presence woven through a shared condition. Seen this way, its global reach takes on a quieter form. It is widespread not because it travels across the surface, but because the environment it requires exists in many places at once. The ocean provides that environment repeatedly, shaping it along gradients of depth and temperature, allowing the same kind of life to exist beneath distant waters that never meet at the surface. This also explains its absence from ordinary observation. The layer it inhabits lies beyond casual reach, below the zones most often explored, and within a range where visibility depends on artificial light that alters the surroundings. The fish remains within that layer, moving through it as part of a system that is rarely intersected directly. So its presence becomes a quiet constant, extended across the globe but concealed by the nature of its habitat. Beneath the Atlantic, beneath the Pacific, beneath the Indian Ocean, the same narrow band of cold water persists, and within it, the same form continues its slow movement, guided by gradients that remain invisible from above, existing not in a place that can be pointed to, but in a condition that circles the earth continuously. Silence at these depths is not emptiness but compression, a narrowing of signals into faint disturbances that pass through water and vanish again. Within that narrowing, every movement carries consequence, not only for what is hunted, but for what hunts in return. The black swallower moves through this space as a predator, shaped by scarcity, yet it does not move alone. No direct observation records what takes it. There are no images, no sequences of pursuit or capture, no confirmed moment where its role reverses. What exists instead is alignment, the overlap of habitats, behaviors, and feeding patterns that trace the outlines of unseen interactions. From these alignments, a structure begins to appear, one built not from single events, but from repeated coincidence across time. Among the most consistent presences in the same depth range is the lancetfish, Alepisaurus ferox, a long, blade-bodied predator that drifts and accelerates through the mesopelagic with little apparent selectivity. Its stomach contents, examined across many specimens, reveal an indiscriminate appetite: fish, squid, crustaceans, anything that can be seized and swallowed. Within that list are species that share the same vertical band as the black swallower, occupying the same cold layer where encounters become possible without ever being seen. Above and below, other forms pass through in arcs rather than residents. Large pelagic predators, swordfish, marlins, certain tuna species descend from brighter waters in controlled dives, pushing down through pressure gradients to reach concentrations of prey that gather beneath the surface layers. These movements are brief, but they intersect the same depths, sometimes exceeding 600 meters, sometimes deeper, placing fast, visually guided hunters into a domain where most life relies on other senses. Further still, the water is crossed by something that does not remain within it at all. Sperm whales descend from the air into the ocean, carrying with them the capacity to reach depths beyond 1,000 meters, and in some cases exceeding 2,000. Their dives trace long vertical paths, guided by sound rather than light, and their feeding leaves a record in the contents of their stomachs, where an extraordinary diversity of deep-living organisms appears, drawn from the same zones the swallower inhabits. None of these records name the black swallower directly in a definitive sense. Instead, they define a field of possibility, a set of predators whose ranges overlap with its own, whose feeding behaviors include species of similar size and form, whose presence is constant enough to shape the evolution of anything sharing their space. The body of the swallower reflects this unseen pressure. Its skin absorbs light that is scarcely present, reducing what little signal might reveal it. Its form minimizes disturbance, slipping through water with limited wake, reducing the pressure traces that other animals might detect. Its sensory focus turns outward through the lateral line, not only to locate prey, but to register movement that does not belong to it. These features do not emerge in isolation. They accumulate over time, shaped by repeated encounters that leave no direct record, only the gradual refinement of traits that persist because they reduce the chance of being found. The absence of observation does not indicate absence of interaction. It indicates a system operating beyond the reach of continuous sight. Across millions of years, something has followed the same thermal band, the same pressure range, the same darkness, intersecting the swallower's path often enough to matter. Each generation carries forward the imprint of those intersections, encoded in structure and behavior, even as the events themselves remain unobserved. So the black swallower moves as both hunter and potential prey, its existence defined by a balance that is never directly witnessed. In the same cold layer that sustains it, other bodies pass, guided by different senses, driven by different needs, occasionally crossing its path in moments that leave no trace beyond the quiet continuation of a lineage shaped by forces that remain largely unseen. Far above the depths it inhabits, sunlight drives a quiet transformation, turning dissolved carbon into living tissue, shaping the base of an immense, drifting system. That surface process feels distant from the cold band where the black swallower moves, yet the two are linked by a nightly exchange that binds the upper ocean to the deep. As darkness settles across the surface, vast numbers of mesopelagic organisms rise from below, crossing layers of water that differ in temperature, pressure, and light. They enter regions where phytoplankton and other primary producers have already fixed carbon from the atmosphere, converting it into organic form. There, in the brief window of night, they feed, taking in that carbon as part of their own bodies. The black swallower moves within this migration, rising into zones where prey is more abundant, where the results of surface productivity become accessible. Each successful capture transfers that carbon again, from one body into another, concentrating it into a form that will not remain near the surface for long. By the time the light returns, the movement reverses. The ascent dissolves into descent, and the entire system begins to sink back toward colder, darker water. With that descent, the carbon moves as well. It is no longer suspended in the upper layers, where exchange with the atmosphere can occur. It is carried downward inside living organisms, transported across gradients that separate surface from depth. The black swallower, with its large and infrequent meals, becomes a particularly efficient carrier in this process, holding within its body a concentrated package of organic material that will be processed slowly over time. Digestion in cold water proceeds at a reduced pace, stretching the conversion of that material across days and weeks. During this period, waste products are released at depth, particles settle, and in some cases, entire bodies eventually become part of the descending flux. Each of these pathways moves carbon further from the surface into regions where it may remain isolated from atmospheric exchange for long periods. This transfer is not a singular event, but a continuous flow, repeated every night across every ocean basin. It forms part of what is known as the biological pump, a system that shifts carbon from the upper ocean into the deep, regulating its distribution across the planet. Without this movement, carbon would accumulate differently, altering the balance between ocean and atmosphere in ways that would reshape climate over long timescales. The scale of this process is difficult to hold in a single frame. The mesopelagic zone contains an immense amount of living mass, estimates ranging from 1 to 10 billion metric tons, dispersed through layers that rarely come into direct view. Compared to this, the annual global catch of fisheries, around 80 million metric tons, represents only a fraction of what exists unseen below. Within that larger mass, countless individual movements contribute to a collective transfer that operates continuously. The black swallower participates in this system without variation, its behavior shaped entirely by the need to locate and process food in an environment where meals are uncertain. Its nightly ascent and descent align with the broader migration, placing it within the flow of carbon that links surface productivity to deep storage. Each cycle of feeding and digestion becomes part of a larger pattern, one that extends far beyond the scale of the individual animal. There is no awareness within the fish of the role it plays. It does not register the chemistry of carbon or the dynamics of climate. Its movements follow gradients of pressure and the signals detected through its lateral line, guiding it through a world defined by physical constraints rather than abstract systems. Yet, through those movements, repeated across generations, it contributes to a process that has been operating for hundreds of millions of years, quietly shaping the balance of the planet. So the descent continues, carrying within it the residue of surface light, transformed into mass and drawn downward into cold, stable layers. Within that motion, the black swallower remains what it has always been, a participant in a system far larger than itself, moving through darkness with a burden that will settle into the deep and remain there. Long after the surface has changed again, the seafloor off Nova Scotia does not descend gently. It breaks, opening into a long, steep-sided canyon carved into the continental shelf, a formation known as the gully. From the surface, nothing marks its presence clearly, only subtle shifts in current and depth charts that hint at a structure reaching downward for nearly 2,000 meters, cutting through layers of water that rarely meet the light above. Along these walls, the ocean arranges itself into vertical bands, each defined by pressure, temperature, and the absence of sunlight. The same narrow thermal range that shapes the movement of the black swallower passes through this canyon, stretching from its upper reaches down toward its deepest contours. The environment does not begin or end at a border. It extends continuously through the space, allowing organisms adapted to these conditions to occupy it without interruption. In 2004, this region was designated as Canada's first offshore marine protected area, a recognition not of what is easily seen, but of what exists within these depths. The designation restricts certain human activities, most notably deep-sea trawling, a practice capable of dragging weighted nets across large vertical and horizontal distances, intercepting animals throughout the water column and along the seafloor. In a system where recovery operates slowly, such disturbances can alter entire communities before their full structure is even understood. Within this protected space, the presence of the black swallower was confirmed in a way consistent with nearly all knowledge of the species, through specimens recovered rather than individuals observed alive. A study conducted in 2024 examined 53 individuals collected from the gully, applying computed tomography to reveal internal structures without dissection. The scans traced the arrangement of vertebrae, allowing researchers to identify the species with precision and to document its occurrence within the boundaries of the protected area. This method reflects the broader pattern that has followed the fish through scientific history. Its form is known through stillness, through bodies that have ceased to move, through imaging that reconstructs what cannot be witnessed directly. Even here, in a region set aside for preservation, its presence is confirmed indirectly. a continuation of the same distance that has defined its study for nearly 2 centuries. The canyon hosts other inhabitants that traverse similar depths. Northern bottlenose whales move through these waters on long dives, descending beyond 1,000 meters in search of prey. Their paths intersect the same vertical range, linking surface and depth through repeated movement, though the exact points of interaction remain unrecorded. The shared space suggests overlap, not through direct observation, but through the alignment of depth, temperature, and feeding behavior. Protection in this context operates without complete knowledge. The black swallower has not been assessed by the International Union for Conservation of Nature. No global population estimate exists, no baseline against which change can be measured, no trend that can be followed over time. Its apparent abundance in some regions coexists with a lack of quantified data, leaving its status undefined rather than secure. The gully stands as a rare instance where a portion of its environment is formally shielded despite this uncertainty. The canyon walls, the cold layers they contain, and the organisms that move within them are granted a degree of separation from direct disturbance. This does not reveal the life that passes through, nor does it resolve the absence of observation, but it preserves the conditions under which that life continues. Within this protected descent, the same patterns persist. The black swallower moves through the thermal band that threads the canyon, rising and falling within its limits, encountering prey, carrying carbon, avoiding what hunts in return. The designation above does not alter these movements. It only ensures that the structure supporting them remains intact, a quiet acknowledgement that even without full understanding, the continuity of such environments holds value in itself. Knowledge of this animal arrives after motion has ended. It is assembled from stillness, from bodies recovered without context, from forms that no longer act, but still retain the structures that once allowed them to. The black swallower has been approached in this way since its first collection, each specimen adding detail while withholding the sequence of events that gave those details meaning. The largest single effort to understand its feeding comes from 337 individuals examined from the Gulf of Mexico, collected during research cruises between 2010 and 2011, and analyzed in a study completed in 2024. Within those bodies, prey remains provided a record of diet, a partial reconstruction of interactions that occurred beyond observation. Patterns emerged through accumulation, repeated across many specimens, forming a statistical outline of what the fish consumes without ever showing how those meals were obtained. A separate advance appeared in the use of computed tomography, applied to 53 specimens recovered from the gully. Instead of dissection, imaging revealed internal arrangements intact, tracing vertebrae, organ placement, and structural variation without altering the bodies themselves. This method extends the amount of information that can be drawn from limited material, preserving what is rare while still extracting detail. Even so, it continues the same pattern, learning from what has already ceased to live. From these approaches, a coherent picture has formed in fragments. The anatomy is understood, from the arrangement of jaws to the expansion of the stomach. The coloration is known, the depth range traced, the temperature band defined. Diet composition has been measured, reproductive traits outlined in broad terms, egg structure described, juvenile stages identified. Fossil relatives extend the lineage backward, anchoring it in a longer evolutionary context. Alongside this, a second list persists, one that does not resolve with additional specimens. Lifespan remains unmeasured. Mating behavior has not been observed. Social interactions, if they exist, are unrecorded. Navigation through a featureless environment remains inferred rather than demonstrated. The frequency of successful feeding compared to fatal overreach is unknown, existing only as an imbalance in the specimens that reach the surface. The sensitivity and effective range of the lateral line are not directly quantified. The transition from juvenile stages near the surface to adult life in deeper layers has never been followed continuously. Whether individuals occupy defined areas or drift through broader currents is not established. These gaps are not the result of oversight. They arise from the conditions in which the animal exists, a space where sustained observation is difficult to maintain. The deep ocean resists long presence, limiting the duration and continuity of direct study. Instruments descend, record briefly, and return. Nets collect without witnessing. Cameras illuminate only small volumes within a vast expanse. The result is a record composed of endpoints rather than processes. Nearly 2 centuries have passed since the first specimen was brought up from depth. In that time, techniques have advanced, instruments have improved, and the amount of collected material has grown. Yet the balance between what is known and what remains uncertain has not shifted as much as might be expected. Each answer reveals additional structure, but also defines the boundaries of what cannot yet be seen. The black swallower exists within those boundaries, its life unfolding in sequences that leave only partial traces behind. The facts that describe it are precise, grounded in repeated examination, but they remain separated from the flow of events that produce them. Between those facts lies a continuity that has not been directly observed, a set of behaviors and interactions that connect anatomy to action. So the understanding of this animal remains anchored in what the dead can provide. Each specimen contributes another fragment, another fixed point within a larger system that continues beyond view. The living process persists in darkness, while knowledge gathers slowly at the surface, shaped by absence as much as by what can be held and measured. The deep ocean long existed as a region shaped almost entirely by internal forces, pressure, temperature, and the slow circulation of water across basins. In recent time, influences from above have begun to extend downward, not as a single intrusion, but as a series of gradual changes that reach into layers once considered distant from surface activity. Among the most direct of these is deep-sea trawling. Industrial nets, weighted and drawn across wide vertical ranges, can descend to depths approaching 2,000 meters and sometimes beyond. As they move, they intersect the same thermal band that the black swallower occupies, passing through the water column with little distinction between species. The swallower is not a target of this activity, lacking commercial value, yet it becomes part of the incidental catch, removed alongside other organisms whose roles within the system remain only partially understood. The scale of this interaction is difficult to quantify, as bycatch records rarely resolve to this level of detail, leaving the number of individuals affected uncertain. Changes in temperature extend the influence of the surface in a different way. As the upper ocean absorbs heat, the structure of thermal layers begins to shift, altering the depth at which specific temperature ranges occur. The band between 4.2 and 8.7 degrees Celsius, which defines the swallower's preferred environment, is not fixed in place. It moves, rising or sinking depending on regional conditions, reshaping the vertical distribution of organisms that depend on it. For a species whose presence is tied closely to this narrow range, such movement introduces a gradual reorganization of habitat that unfolds across large spatial scales. Oxygen availability follows a related pattern. Warmer water holds less dissolved oxygen, and regions of low oxygen concentration, known as oxygen minimum zones, have been observed to expand within the mesopelagic. As these zones grow, they compress the layers where oxygen remains sufficient for sustained activity. Organisms adapted to specific concentrations may find their viable range narrowed, adjusting their movements within a thinner band of water. For the black swallower, which already operates within defined limits of temperature and depth, this additional constraint further shapes the space it can occupy. Material introduced at the surface now reaches depths once considered isolated. Microplastics have been documented at levels exceeding 6,000 meters, carried downward through sinking particles and biological transport. Within the mesopelagic, these fragments enter food webs indirectly, passing from smaller organisms into larger ones through feeding interactions. The swallower, consuming prey drawn from this system, becomes part of that pathway. The extent of this incorporation and its physiological effects remain under investigation, with current understanding based on detection rather than long-term observation. Across these influences, a consistent feature remains: the absence of a clear baseline. The black swallower has not been assigned a formal conservation status, not because its condition is known to be secure, but because the data required to assess it are incomplete. There is no comprehensive estimate of population size, no long-term record of change, no established trend against which recent pressures can be measured. The species exists within a range that is vast, dispersed, and rarely sampled in a consistent way. This absence does not indicate stability or decline. It reflects the difficulty of measuring systems that operate beyond continuous observation. The pressures introduced from the surface are measurable in their own domains: temperature, oxygen, material transport, but their combined effects on a species like the black swallower unfold within layers that are only intermittently accessed. So, the environment continues to shift, slowly adjusting the conditions that define where the swallower can exist. The fish moves within those adjustments, following gradients that change over time, its behavior guided by the same sensory systems that have shaped it across millions of years. Above, the drivers of those changes persist, extending their influence downward, altering the structure of a habitat that has long remained beyond direct view, yet is now increasingly connected to the processes of the surface world. Across the depths where light does not reach, variation does not fade, it intensifies. The conditions that define this region do not narrow possibility, they refine it, pressing each lineage towards solutions that would seem improbable elsewhere, yet remain consistent with the same underlying constraints. The black swallower exists within this field of solutions, one expression among many that have taken shape under the same persistent forces. Scarcity governs the pace of life here. Food arrives irregularly, in fragments or in sudden abundance, separated by long intervals where nothing moves within reach. Darkness removes visual certainty, replacing it with gradients of pressure and chemistry. Cold slows internal processes, stretching time between intake and use. Pressure alters the physical limits of structure, shaping tissues and molecules alike. Isolation limits interaction, separating populations across distances that are not measured horizontally, but vertically, within layers that rarely mix. Within this framework, different strategies emerge, each tuned to a specific way of meeting the same requirement, acquiring enough energy to persist and reproduce. The anglerfish approaches this through attraction, extending a bioluminescent lure from its body, creating a point of light that draws prey inward. In some forms, reproduction itself becomes part of the adaptation. Males reduce to minimal structures that attach permanently to the female, merging circulatory systems and remaining as a constant source of gametes, an arrangement that ensures reproduction in a space where encounters are uncertain. Elsewhere, the gulper eel expands the limits of intake through structure, its jaw opening to an extent that allows the capture of prey far exceeding its resting proportions. The act is singular and expansive, a brief moment of extreme extension followed by a long period of processing. This approach aligns with the same pattern seen in other deep-sea organisms, infrequent feeding events balanced by prolonged digestion. The barrel-eye fish adopts a different orientation, its head forming a transparent dome that encloses upward-facing tubular eyes. These eyes pivot within the fluid-filled structure, scanning the faint residual light that filters from above, detecting silhouettes that pass against that dim background. The adaptation is not one of expansion, but of perception, refining the ability to locate prey within a limited visual field. The viperfish presents yet another configuration, its elongated fangs extending beyond the closed mouth, forming a set of fixed points that intercept fast-moving prey. Here, the solution emphasizes capture through reach and retention, ensuring that once contact occurs, escape is unlikely. Each of these forms addresses the same underlying condition, the need to secure energy in an environment where opportunity is rare. The differences lie in how that condition is approached, through attraction, expansion, perception, or interception. None of these strategies operates in isolation from the constraints that produce them. They are shaped by the same gradients of temperature, pressure, and resource availability that define the deep ocean as a whole. The black swallower occupies a position within this spectrum, extending its capacity through a combination of structural flexibility and commitment to capture. Its approach does not refine a single aspect, but broadens the range of what can be taken, accepting prey that would exceed the limits of most comparable organisms. This places it at one end of the possible solutions, where risk and reward are closely aligned. Viewed together, these adaptations form a coherent pattern rather than a collection of anomalies. The deep ocean functions as a continuous setting in which similar pressures produce diverse yet consistent outcomes. Over millions of years, these outcomes accumulate, each representing a stable response to the same set of constraints. In this sense, the environment does not generate unpredictability, it generates precision. Each form reflects a pathway through the same problem space, shaped by the limits imposed by darkness, cold, and scarcity. The black swallower remains one of these pathways, distinct in its method, yet entirely aligned with the logic that governs all life within this domain. Before any measurement, before any instrument records a signal or a specimen is brought into view, there remains a boundary that observation does not cross. It is not a boundary of distance or depth, but of perspective, the separation between what can be described from the outside and what unfolds within a living body as it moves through its own environment. The black swallower exists entirely on one side of that boundary. Its actions can be reconstructed, its structures mapped, its environment measured in gradients of pressure and temperature, yet none of these fully resolve what those processes are like from within. The signals that guide it, the changes it responds to, occur without language or representation, carried through tissues that convert movement in water into patterns of neural activity. A disturbance passes through the surrounding fluid, a minute shift in pressure, and along the length of its body, the lateral line registers it as a sequence of impulses, each pore contributing to a composite pattern that defines direction, distance, and motion. There is no image associated with this, no outline of a body against a background. Instead, The world arrives as variation, as pulses that differ in intensity and timing, forming a structure that can be acted upon. The distinction between one kind of prey and another is not visual but dynamic, encoded in how water moves when displaced by muscle and fin. A lanternfish produces one signature, a snake mackerel another, each pattern shaped by size, speed, and form. These differences are sufficient. They allow orientation, approach, and engagement without ever resolving into a picture. When contact occurs, the sequence continues without interruption. The jaw engages as a mechanical response, structures moving along paths defined by bone and ligament. The ratcheting motion progresses, alternating sides, drawing the prey inward through a series of incremental advances. Each movement follows from the last, coordinated through neural circuits that do not require reflection or evaluation. The process unfolds as it has been shaped to unfold, step by step, until the prey is contained. Within the body, another transformation begins. The stomach expands, tissue unfolding to accommodate what has been taken in. Stretch receptors respond to the increasing volume, signaling the state of fullness, while chemical processes initiate the gradual breakdown of the prey. The surrounding water remains cold, maintaining a pace that extends digestion across long intervals. During this time, activity reduces, movement becomes minimal, and the body maintains position within its preferred depth, guided again by gradients that are sensed rather than seen. The vertical movements that structure its existence occur in the same manner. As it rises and descends, the changes in pressure and temperature are continuous, forming a profile that can be followed without reference to landmarks. The absence of light does not remove orientation. It replaces it with other forms of information, ones that remain stable across the vast distances of the ocean column. All of this can be described in terms of function and mechanism, yet the interior aspect remains inaccessible. There is no direct account of how these signals are integrated, no description of whether they cohere into anything that could be called a unified state. The nervous system of the black swallower is sufficient for its behavior, but its organization differs from those systems more commonly associated with complex cognition, leaving open the question of what, if anything, accompanies its actions beyond the execution of those actions themselves. This uncertainty does not arise from a lack of detail in measurement, but from the nature of the problem. The processes can be traced, the pathways identified, yet the presence or absence of experience cannot be derived from structure alone. It remains a possibility that extends alongside the known, neither confirmed nor excluded. What can be said with certainty is that the sequence of sensing, movement, capture, and digestion has persisted over spans of time that exceed any human observation. It continues in the same conditions that shaped it, within an environment that remains largely unchanged in its fundamental properties. Whatever accompanies these processes, if anything does, is part of that continuity, unfolding in the same cold, dark water, carried forward as one more expression of life, operating within the limits and possibilities of this planet. There is a tendency, when confronted with an organism that fails in such a visible way, to treat the failure as the defining feature. Yet, in this case, the visible moments are only a narrow slice of a much larger pattern, one that extends across millions of years and countless unseen encounters beneath the surface. The black swallower does not persist because it avoids risk. It persists because its particular balance of risk and reward yields, over time, more continuations than endings. The structure of that balance is embedded in its anatomy. The inward-angled teeth, responsive to pressure yet resistant to reversal, create a system that favors capture above all else. The expanding stomach, capable of unfolding far beyond the apparent limits of the body, allows it to convert rare opportunities into extended periods of survival. These features operate together, forming a single integrated response to an environment where food arrives unpredictably and may not return for long intervals. In such a setting, hesitation carries its own cost, one that accumulates quietly in the absence of intake. What appears as excess is, in this context, proportionate. The ability to secure prey larger than the predator itself is not an anomaly, but a solution tuned to scarcity. Each successful event provides a reservoir of energy that sustains the fish through weeks of minimal activity, its metabolism adjusted to extract and conserve what it has obtained. The intervals between these events are not empty spaces but essential components of the cycle, shaped by temperature, pressure, and the slow pace of biochemical processes in cold water. The failures, when they occur, follow from the same mechanisms. A prey item that exceeds the limits of digestion initiates a sequence that the body cannot interrupt. The processes that normally convert tissue into usable energy begin to diverge, decomposition advancing in parallel with digestion. Gas accumulates, buoyancy shifts, and the surrounding conditions change in ways the fish is not structured to counter. This progression is not a separate pathway, but an extension of the same pathway that, under different proportions, leads to survival. From the perspective of evolution, these outcomes are not weighted equally in isolation. What matters is the aggregate effect across many individuals and many generations. If the strategy yields a sufficient number of successful cycles to sustain the lineage, it is retained. The presence of recurring failure does not negate the effectiveness of the design. It defines its boundary conditions. Within those conditions, the system operates as intended. Our view of the black swallower is shaped by a bias toward what reaches us. The specimens that surface, the ones preserved and studied, are those that have crossed beyond the viable range of their adaptations. They provide detailed information about structure and potential, yet they represent a subset defined by breakdown rather than continuity. The majority remain unobserved, their lives unfolding entirely within the parameters that support them. Those unseen individuals carry out the same sequence without interruption. They encounter prey that falls within the tolerances of their bodies, engage, consume, and then descend, entering the long interval of processing that follows. Their presence is not marked by emergence or collection. It is expressed through persistence, through the quiet continuation of a pattern that rarely intersects with human observation. In this way, the central paradox resolves into a principle that extends beyond a single species. Adaptations are not separate from their limits. The feature that enables survival also defines the edge at which survival gives way to failure. The black swallower exists precisely at that edge, its design neither excessive nor deficient, but aligned with the demands of its environment in a way that has endured across geological time. The animal that comes into view, distended and still, offers a clear image of that edge. The animals that remain below it do not present themselves, yet they are the ones that carry the pattern forward, continuing the sequence of capture, descent, and slow transformation in the dark water, where the conditions that shape them remain unchanged. At a certain point, the scale of what surrounds this animal begins to shift the frame of the story. The black swallower is no longer the center, but a single thread moving through a vast, continuous layer that wraps the planet in darkness just beyond the reach of ordinary sight. The water above it reflects sky and weather, familiar and constantly observed, while below that thin boundary another ocean extends, structured by gradients rather than light, inhabited by forms that rarely intersect with the surface world. This region, the mesopelagic, occupies most of the habitable ocean by area, a band that stretches across basins and between continents, following depth and temperature rather than geography. Within it, life accumulates in numbers that are difficult to hold in a single image. Estimates suggest between 1 and 10 billion metric tons of biomass moving through this layer, rising and descending in coordinated cycles that have no parallel on land. These movements occur without announcement, without visible trace from above, yet they represent one of the dominant biological processes on Earth. The black swallower moves within this system as one participant among many. Its nightly ascent places it in proximity to a shifting field of organisms, each following its own pattern, each contributing to the broader exchange of energy and matter that defines the zone. The water column becomes a space of interaction structured by depth and time, where encounters occur not in fixed locations, but along pathways that repeat with the turning of the planet. Despite the scale of this activity, direct observation remains limited. Human exploration has reached extreme points, descending to the deepest trenches and traveling beyond the atmosphere, yet the continuous monitoring of this midwater region has not been achieved. The tools that enter it, remotely operated vehicles, and instrumented platforms, illuminate small volumes for short durations. They capture fragments, moments that hint at broader patterns, but do not resolve them fully. Within those fragments, thousands of species have been recorded, many described only recently, others known only from specimens collected without context. The behaviors that structure their lives, feeding, reproduction, interaction, remain largely inferred. The black swallower, with its long history of study, stands among the more documented inhabitants, yet even here, the outline exceeds the detail. Its presence is established across oceans, its anatomy understood in precision, but the continuity of its life remains unseen. This imbalance between knowledge and scale defines the mesopelagic as a kind of interior space within the planet, one that is physically connected to the surface, yet functionally distinct. It is not distant in the way that other worlds are distant. It lies directly beneath waters that can be crossed in hours, accessible in principle yet resistant in practice. The conditions that preserve its inhabitants, pressure, darkness, and depth, also limit the methods by which they can be observed. Every point within this layer contains activity that has not been recorded. Movements occur without witness, interactions complete without leaving traces that persist to the surface. The absence of observation does not imply absence of complexity. It indicates only that the processes unfold beyond the current reach of continuous study. The black swallower, moving through this environment, participates in patterns that extend far beyond its own scale, patterns that shape the distribution of energy and matter across the ocean. Seen from this perspective, the animal becomes both specific and representative. Its particular adaptations are one solution among many, its life one instance of a broader system that operates across the globe. The darkness it inhabits is not empty, but structured, filled with gradients and movements that define a second ocean layered beneath the first. This deeper ocean persists regardless of attention. It continues its cycles, maintains its populations, and supports interactions that remain largely unrecorded. The surface world passes above it, marked by light and weather, while below, in cold water that changes slowly and holds its conditions across vast distances, life continues in forms that have yet to be fully seen. long before any shoreline carried the imprint of human passage, before tools, before language, before even the earliest ancestors that would one day look upward and name the sky. This lineage had already settled into its pattern. It existed within a world that required no light to define it, no surface reference to orient it, only gradients of pressure and temperature that have persisted with remarkable stability across geological time. 13 million years extends beyond ordinary scales of comparison. In that span, continents shifted incrementally, mountain ranges rose and eroded, and entire climates reorganized themselves in cycles that reshaped the surface again and again. The Himalayas continued their slow ascent, the Mediterranean basin experienced isolation and return, and polar ice advanced and retreated in rhythms that defined epochs. Across these transformations, the deep ocean remained comparatively constant, its darkness unbroken, its temperature held within narrow limits, its pressure unchanged except by depth. Within that continuity, organisms adapted not to rapid change but to enduring constraint. The black swallower's ancestors occupied the same thermal bands, the same vertical ranges, responding to the same scarcity that defines the mesopelagic. The strategy that now appears extreme was, even then, a direct response to those conditions. There was no need for frequent reinvention, no pressure toward visible alteration, only the gradual refinement of mechanisms that already aligned with the environment. As surface ecosystems reorganized, producing new forms and extinguishing others, the deep remained insulated from many of those shifts. The absence of light created a buffer against fluctuations driven by solar input, and the immense volume of the ocean stabilized temperature over long intervals. In this setting, evolutionary change proceeded at a pace dictated less by external disruption and more by the slow accumulation of variation within a stable framework. The black swallower, as it exists now, reflects that history. Its form is not recent in any meaningful sense. It represents a configuration that has persisted because it continues to function within the same constraints that shaped it. The movements that define its life, the ascent, the encounter, the descent, would be recognizable across vast stretches of time. Generations separated by millions of years would occupy the same layers, respond to the same signals, and complete the same sequence of actions. Human history, by contrast, occupies only a narrow interval within this timeline. Entire civilizations rise and fall within periods that, on a geological scale, pass almost without trace. Languages emerge, transform, and disappear. Technologies develop and are replaced. These processes unfold rapidly at the surface, creating a sense of constant change that contrasts sharply with the continuity below. The black swallower exists outside that rapid turnover. Its existence does not intersect with the events that define human chronology. It does not register the rise of cities or the alteration of coastlines. Its world is structured by factors that remain largely independent of those developments. Pressure increases with depth, temperature stabilizes within a narrow band, and darkness persists regardless of conditions above. This separation does not imply isolation in a physical sense, but rather a difference in the scale and type of change experienced. The deep ocean evolves, but it does so within parameters that shift slowly, preserving conditions that allow certain strategies to remain viable over immense durations. The black swallower's continued presence is evidence of that stability, a living indication that some environments favor persistence over transformation. In this way, the animal becomes a measure of time not through its own awareness, but through its continuity. It carries forward a pattern that has endured through epochs, maintaining a relationship with its environment that has required little alteration. The cold, the pressure, the absence of light, these have remained sufficiently constant to support the same fundamental approach to survival. What appears unchanged is not static, but sustained. It is the result of conditions that hold steady across spans of time that exceed ordinary perception, allowing life to settle into forms that can persist without interruption. The black swallower moves within that continuity, its existence a quiet extension of a history that began long before any record and continues without need for acknowledgement. It returns, in the end, to that single image drifting in warm surface water, a small, dark body carried by currents it was never meant to meet, its form altered beyond recognition by the process that brought it there. What once appeared as an anomaly now settles into place as the visible endpoint of a sequence that began far below, in conditions that remain unchanged by the light above. On that night, the ascent followed the same path traced countless times before, a movement guided not by sight, but by gradients and signals carried through water. Somewhere along that rise, a pattern of vibration resolved into opportunity. The lateral line registered motion, direction, the distinct rhythm of a fish moving through the same column. There was no hesitation in the response. The jaws engaged as they were shaped to engage, the teeth aligning and locking, the mechanism closing into its single direction. The process that followed unfolded with steady precision. The body advanced along the prey, alternating movement, drawing it inward through a sequence that has no reverse. What entered the mouth did not leave it. The stomach received what the jaws delivered, and as it did, it began to expand, layers of tissue unfolding to accommodate the mass within. The descent came after, carrying both predator and prey back into colder water, where time stretches and processes slow. Within that cold, the conversion began, the long interval in which structure becomes energy. Under ordinary proportions, this is where the cycle stabilizes. The fish remains nearly still, its metabolism reduced, its body committed to extracting what it has taken. The surface recedes, the darkness resumes its full presence, and the sequence continues toward completion. In this instance, the balance shifted. The size of the prey extended beyond the pace at which digestion could proceed. Within the enclosed space of the stomach, chemical processes moved along a different path, producing gases that accumulated with no means of release. Buoyancy altered gradually, then decisively, and the surrounding pressure began to fall as the body rose, carried upward by forces that do not negotiate. The ascent through warmer layers introduced changes the fish was not structured to counter. Expansion followed decompression, each increment amplifying the last. The boundary between the deep and the surface closed, and the fish crossed into a world defined by light and air, conditions that had not shaped its adult form. There, the sequence ended in visibility. The internal, now external, the hidden made briefly accessible. Elsewhere, beyond the reach of that surface, the same sequence proceeds without interruption. Individuals complete the cycle within its viable range, their meals proportionate to the limits of their bodies, their descent leading not to exposure but to continuation. They return to the depths with what they have secured, entering again the long interval, where energy is drawn out slowly, where movement is minimal, where survival is extended across time. When that interval ends, the pattern resumes. The body reduces to its lean form, the internal space clears, and the sensory system returns to its quiet attentiveness. Signals pass through the water, and the animal responds, rising again along pathways that have no markers but remain consistent through repetition. The ocean above and below maintains its structure, and within it, the cycle continues. Across the span of years that define this lineage, this sequence has repeated without interruption. It has carried individuals through success and failure, through continuation and ending, without altering the fundamental relationship between body and environment. The deep ocean holds its conditions, and within those conditions, the same set of actions unfolds. The image at the surface remains as a record of one outcome among many. It is complete in itself, yet it points beyond itself to the larger pattern that persists unseen. Beneath the water, within layers defined by cold and pressure, the same movements are occurring now, as they have for millions of years. Each one a continuation of a life that operates entirely within the limits that shaped it. And within that continuity, the sequence does not conclude. It carries on in darkness, in stillness, in the steady repetition of a form of existence that remains aligned with its environment, sustained by conditions that have not required it to become anything other than what it already is.